rid of it, even though it's a player drawing. But welcome to, I think this is like the 21st revision stream now. And to be honest, I don't know what I want to revise today. I think it's probably worth having a look over, what we've been covering in school recently. Because it's all of the kidneys. It's osmoregulation, basically everything to do with the kidney nephrons. So I think what we'll do is we'll spend 5-10 minutes going over that quickly, seeing what we can remember, and then we can probably just try some past paper stuff, see how far we get. So let's get into this. So the kidney nephron. Firstly, I guess it's important to understand the parts of a kidney. So what we have is, starting from the top, you have the cortex, the medulla, and then I forget what you call it. The... Oh no, what's the word? No, what is it? The inner bit of a kidney, the white bit, that kind of... Oh, I don't recall what it is. Pelvis, the pelvis, I think. I think it's a pelvis. Okay. So that is the structure of the kidney itself. Okay. Gonna move that back a little. That is the structure of a kidney. The nephron starts off like this, and I'm going to try and draw it just from memory. I'm going to kind of draw it. That is kind of how I remember it. So we have our loop of Henle. We have our, this is our distal, no, what's it called? Oh my god, why can't I remember the word? DCT? Distal convoluted tubule. That's it. And this is going to be the something convoluted tubule. Is it PCT? I don't remember. I don't recall the word. The distal and something convoluted tubule. That's the loop of Henle. You have the descending and ascending limb. This is going to be the Bowman capsule. That is going to be the glomerulus. I can't pronounce it. The glomerulus. The glomerulus. I don't know how you pronounce it. But that is the nephron. And this is obviously going to be the duct. I don't recall what this is. It's for something convoluted tubule. I don't recall, because I know that's distal, so this has to be some kind of mashup of the words. Proximal, that's it. Proximal convoluted tubule. Okay. So that is the kidney nephron. Okay, So, 3 stages of filtration in the kidneys. First stage is going to be ultra filtration. Now, if you get asked about this in an exam, you can get 3 easy marks just for doing the following. Number 1, you specify there is a high hydrostatic pressure. This pressure is coming from the something arterial, sorry. The something... It's from the something arterial. Is it like the eruvant arterial or something? I don't recall. It's the something arterial. Again, just learn this topic. There's going to be some words that I don't recall. Ultrafiltration. High hydrostatic pressure in the arterial. Again, I don't recall what it is. And this forces out, and you need to be able to name here, things like ions, water, I think glucose, urea. Just to be safe, I'm going to replace glucose with amino acids for now, even though I know it is one. And they are forced out via the capillary pores. So, there you go, ultrafiltration, a high hydrostatic pressure in the something, I forget the word, arterial, forces ions, water, amino acids, urea, through the capillary pores and into the Bowman's capsule. So that is ultrafiltration. So I'm going to draw a very crude diagram of this, because the gimmick you need to understand here, guys, is the idea that the capillary pores are small enough so that things like proteins and red blood cells aren't able to enter. So we draw something like this for a protein. It can't enter, but things like ions are able to enter. So, that is ultrafiltration. And again, I don't recall what arterial it is. I feel like it's something like the afferent arterial. I feel like that's it. That is literally ultrafiltration, summarized in a very simple diagram. Next is selective reabsorption, which is literally co-transport, okay? Co-transport, that is literally what it is. This is in the proximal convoluted tubule. This is one of the easiest processes that you could get asked like a 5 marker on. It is really easy to understand. So imagine... Okay, we're going to have to draw quite a bit here. So, this process is very simple to understand. It is literally just co-transport, which you should know off by heart already. And this is selective reabsorption. So, selective reabsorption. Firstly, we have the NAK pump, the sodium-potassium pump, which pumps out sodium via active transport. Because there is now less sodium inside the epithelial cell, sodium from the, I don't specifically know what this will be, from the filtrate, sorry, this is the filtrate, that's the capillary, that is the epithelial cell. So sodium from the filtrate is going to enter via facilitated diffusion. It will also drag with it glucose molecules through a symporter. And from there, the glucose is just going to leave via facilitated diffusion, and the sodium will once again travel down the concentration gradient out of the sodium potassium pump. So, glucose is going against its concentration gradient, but it doesn't require ATP due to the fact that it's being brought through with sodium. So literally, this is just playing with concentration gradients. It's very easy to understand. And finally is going to be the loop of Henle, which I'm going to draw. I'll draw it three-dimensional, actually. We'll draw it three-dimensional. So, that's the loop of Henle, the distal convoluted tubule, the collection duct. Okay. Important gimmick number one. This side is impermeable to water. No. This side is permeable to water. This side is impermeable. Good to know? Good to know. Stage one is in the filtrate. Sodium and chloride ions are going to be moved via, I think it's going to be active transport. I think it's active transport of sodium ions and chloride ions, I think. Now, because the solution inside the cell, loop, because the filtrate is now going to be more dilute, no, okay. Wait, no. Hold on, I'm remembering. Okay. This is such a difficult process to put into words, but I understand it. Okay. Stage 1, Stage 2, Stage 3. Stage 1, we have this box full of water and a lot of ions. If we remove the ions, it becomes smaller. What I'm trying to convey here is there's a lower water potential. So, this lowers water potential in the filtrate. So, water is going to travel via osmosis. This is going to be stage 3. Water is going to travel via osmosis into the permeable side, the descending loop of Henle. Now, because we've increased the amount overall, we are then going to start to get facilitated diffusion of Na and Cl via facilitated diffusion and eventually active transport. And overall, this is going to make the solution more dilute. Oh, I don't recall. It'll make the solution more dilute, which will mean that water will once again leave. Water will leave via osmosis. I feel like I've done this wrong. Hold on. I just want to check the document to make sure that I am not waffling here. Because again, it's so easy to make mistakes in biology, even more difficult to understand where you've gone wrong. Water moves out of the limb. They're actively transported out of the limb, like we said. water moves out, so I said water moves in. That was the first mistake, and that's gonna fuck up everything, so let me change that. Let me see if I can do it from here, just from memory then. So, stage 1, active transport. Active transport of the ions. Wait, so why would water leave? Is it because the tissue is going to be more concentrated? Maybe? I don't actually know. Yeah, the fluid has a, oh, it's the fluid. Okay, I was right. Yeah, the fluid. Yeah, because obviously that makes sense. I guess this probably isn't the best way to plot this out. I guess in a way we could kind of plot it like this. You have your ascending loop, you have your descending loop. Sorry, that's going to be the descending loop. That's going to be the ascending loop. Just imagine this is the tissue. So first thing, the ions are going to get pumped out into here. That is obviously going to cause this solution to now be more concentrated with ions. As a result, water is going to move out via osmosis, which will, create an equilibrium, balance it out. Then the same thing is going to happen. Ions are going to move out via facilitated diffusion and then active transport, which means that now there are barely any ions in here, which means that it has a very high water potential overall, so water is going to keep moving out. So what I wrote here is, to begin with, the fluid has a low water potential. Then the filtrate becomes very concentrated, and then the filtrate has a high water potential. So it's a bit difficult to wrap your head around this. I don't know what you would need to say in an exam, because I tried looking for mark schemes on osmoregulation. I couldn't find a single AQA level mark scheme related to osmoregulation. So I don't know what they would want you to say, to be completely honest. I don't know. The way I would write it is I would start by saying active transport of Na plus and Cl minus ions from the ascending limb into the interstitial fluid. From there, I would say that the water moves from the descending limb into the fluid down the water potential gradients. Then I would say that it's active transport, facilitated diffusion, then active transport of the ions into the fluid, which will make the filtrate overall way more dilute, which will then, it's difficult to explain, but I know what I'm saying. It's literally like you're going back and forth. It literally is. You're going back and forth. This makes the water potential in here very low, so water moves out. But because water's moved out, it's now concentrated, so ions move out. And now it has a high water potential, so water moves out. I think in an exam, you could easily get that. You could easily get the concept of that very well in an exam if you spent maybe 5 minutes writing down a few notes before you started. Now we can quickly talk about ADH, which is a hormone. It is secreted in the pituitary gland. It is detected by osmoregulators in the hypothalamus, Thalamus? I don't know. The whole gimmick with ADH is insanely simple, to be honest. It is incredibly simple to understand. We have cells here lining the ducts, the collection ducts. Look, I'm half-arsing it because it's that simple. These are receptors. What's going to happen is our ADH is going to bind. It's going to cause aquaporins to fuse with the membrane, basically giving it more channels, which will then allow water to travel through those channels more easily, which will obviously produce less urine, or will produce more concentrated urine. It's the same gimmick as insulin. When insulin binds with cells, receptors on the cells, it causes channel-carrying vesicles to fuse with the membrane, giving more glucose channels, which allows for an increased amount of glucose to enter the cells, which overall reduces the blood glucose concentration. So, that's everything that I need to really cover. Let's do the second messenger model. I think that's what you call it. Second messenger system. Yeah, okay. This is literally just the effect of adrenaline. So we have a cell here. I'm drawing it square, even though it's not a plant cell, because I don't care. This is a receptor. We have adrenaline binding to the receptor on the cell surface membrane. This forms an adrenaline, I think it's an adrenaline receptor complex. Yep. This is then going to change the shape of this transmembrane protein. Because it changes shape, it is going to cause the activation of what's it called? Actis? No. I'm gonna have to cheat. Adenal. Okay, I was on the wrong idea there. Wrong idea. Adenal cyclase. Oh, I'm just gonna put AC. Adenal cyclase. Adenal cyclase causes, what does it do? It converts ATP to cAMP. ATP to cAMP, which cAMP causes the activation of protein kinase. And protein kinase converts glycogen to glucose. So, main gimmick you need to understand from that is it forms a complex just like the ES complex, just like the antibody-antigen complex. Just use the word complex, you're likely going to get a mark. It activates adenyl cyclase. Yeah, adenyl cyclase. ATP is converted to cAMP. cAMP activates protein kinase. Protein kinase converts glycogen to glucose, which is glyconeolysis, I think. Or glycogenolysis, I think. I don't remember. So, we have been streaming for about 25 minutes. I'm going to go and have a look. Maybe we can do 1 or 2 exam questions. Because, yeah, it's getting a tad late. I know it is a beautiful day today. It's getting to that time now where it's getting a lot brighter in the evening, which is great for me because it means I can do these streams a lot. I'm aiming to try and do one of these streams every day now, even if all I'm doing is going over some generic shit that, like, I know very well. it's all revision at the end of the day. There were some things in a past paper which I really struggled with. One of them was the definitions. So I want to quickly write down a few and try and remember. Things like population, community, species, species richness, ecosystem, community. I've already wrote that. I don't recall half of these, so let me try and give a little guess. I know species richness is the number of different species in a habitat. I know that species is two similar organisms that can reproduce to have fertile offspring. Community, I don't recall. Population, I'm going to guess, is the number of the same species, the same organism in a given area. I don't know. That's what I mean. I don't know these definitions at all, because the mark scheme is very pretentious when it comes to these kind of definitions. It does not want you to use species when it's not on about it. It doesn't want you to talk about anything. Population, yeah, all organisms of the same species in a habitat. So yeah, all organisms of the same species in a habitat. Community is all the populations of different species. I remember my little acronym now. It's PCS SDs, or PCs SDs, because it's populations, community, species, same, different, similar. Ecosystem is a community of different components interacting. So, literally just abiotic and biotic factors. So, PCs and SDs, I'll remember that. Ecosystem is AB. That's literally it. A-B-E. Okay. I'm going to go pull up a past paper. Let's see if we can. Let's see if we can find out some stuff. I guess we can do the 2022. Okay. Actually, I want to try a paper 2. I want to try a paper 2. Let's try the 2023. Let's see if there's any questions. Sorry, the 2022. Okay, a word fill for 2 marks. During photosynthesis, plants produce something compounds which contain carbon. Is that going to be inorganic compounds? or organic compounds, I'm going to say, because inorganic ions don't contain carbon. So we'll say organic. 2. Such as carbohydrates, lipids, and proteins. Most of the sugars synthesized by the plant are used in, I'm going to say, respiration. The rest are used to make groups of biological molecules. These molecules form the biomass of plants. Biomass can be measured in terms of mass of something per given area. Chemical energy can be estimated using no idea. I'm going to guess something like kilojoules. And in terms of mass of waste, maybe? Mass of carbon. So I would have gotten a mark because I got 2 of those. But again, we haven't done that yet. Describe the light... The Calvin cycle. The Calvin cycle. There's 6 marks. Okay. Can we do this? Can we do this? Can we do this? Can we do this? I don't even know what the first step is. I can't remember. I know, it's not pyruvate. Shit, what is it? Rubisco. RUBP and Rubisco. Rubisco is the enzyme. Fuck off. Rubisco is the enzyme which combines something and something into RUBP. That then becomes a 5-carbon compound. No, is that the Krebs cycle I'm thinking of? Would it not split into TP? Would it split into GP, TP, 2TP, and one of them goes off to be used as glucose, and then it comes back together as a 5-carbon compound, which combines with Rubisco. Okay. I don't remember. The only thing that I'm going to say is that a 1-carbon compound, because it's photolysis, the light-dependent reaction. What are the products of the light-dependent reaction? I don't remember. Right, let's just check. I can't be bothered. CO2 combines with RUBP, which produces 2GP. Okay, so it was a single carbon compound. I was right. It's CO2. So I was right. That was probably a mark. A single carbon compound, CO2, combines with RUBP, and that gives you 2GP. Using Rubisco. We back? Okay, so CO2, a 1-carbon compound, combines with RUBP, a 5-carbon compound, catalyzed by Rubisco, gives you 2GP. Or in other words, literally just GP. Cool. Next, I'm going to try and do the rest on my own. What happens for GP to become TP? I'm going to say maybe it's a reduction, which produces oxygen. Maybe. And that leads to those becoming TP. Then I'm going to say one of the triose phosphates becomes a 2-carbon compound, because one carbon is being used to synthesize glucose, and the rest are recycled into the 5-carbon compound IUPP. So, GP reduced to triose phosphate. Should have specified it uses NADH, which I forget exists. NADP, sorry, NADH is for the Krebs. It uses energy from ATP. Triose phosphate is converted into glucose. Triose phosphate converted into glucose. Or IUPP. Okay, so let me try and just sum this all up. The Krebs, not the Krebs cycle, the Melvin-Calvin cycle. So, to begin with, we have a 5-carbon compound which is called IUPP. IUPP, a 5-carbon compound. It is going to combine with a 1-carbon compound, CO2, in order to become a 6-carbon compound. But actually, instead, it's just going to become GP. This is catalyzed, this whole bit here is catalyzed by an enzyme Robisco. The GP and GP are going to be reduced by is it NADH reduced? NADP, sorry. NADPH. And that's going to become TP and TP. The TP, one of them is going to become recycled into a hexa sugar, whereas all the others are going to become there. So that is the Calvin cycle. 5-carbon compound and a 1-carbon compound combined together via Robisco to become 2GP. The 2GP are reduced by NADPH in order to become 2TP, which requires energy from adenosine triphosphate, should specify. I think that that's pretty simple, to be fair. I like the fact that I remembered it. That is a healthy sign. I remembered IUBP, I remembered Robisco, I remembered that it involved reduction, I remembered that it went GP to TP and then back to 5. Should we quickly cover the Krebs cycle? Yeah, because that is a bit different. You start off with follow it. Okay, let's just start from the link, because that's pyruvate. Is pyruvate a 3-carbon compound? I think it is. We'll just put 3-carbon. Pyruvate, 3-carbon compound, is reduced via NAD into acetate, which combines with an enzyme to become acetyl coenzyme A. From there, it then enters the Krebs cycle. It's a 2-carbon compound. It then combines with a 4-carbon compound to become citrate, which is a 6-carbon compound. It becomes a 5-carbon, then a 4-carbon, and then it goes back. Throughout this process, we have the production of ATP twice, the reduction of NAD, the reduction of NAD, the reduction of FAD. This is called decarboxylation. Let me just check. So yeah, the 2-pyruvate become acetate. It's reduced by NAD, or NAD is reduced, which causes... Okay. Acetyl combines to become citrate, so 2 marks. Decarboxylation of citrate into a 5-carbon compound, and the reduction of NAD, 1 mark. Decarboxylation of a 5-carbon compound into a 4, and reduction of NAD, so maybe wouldn't have gotten a mark. Gain of ATP, reduction of FAD, and NAD. Okay, so this is somewhat similar then. It is 2 carbon plus 4 carbon equals 6 carbon. We're going to have 6 carbon going into 5 carbon and NAD into NADH. 5 carbon going into 4 carbon, NAD into NADH. And finally, we are going to have FAD going into FADH, NAD into NADH, and just ATP, or ADP into ATP. So that is it. 2 carbon plus 4 carbon into citrate, citrate into 5 carbon, NAD into NADH, 5 carbon into 4 carbon, NAD into NADH, ADP and PI into ATP, FAD, FADH, NAD, NADH. All you have to do is specify reduction, 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 decarboxylation, gain of ATP. Nice. So that might have been an easy 6 marks, honestly depends. Two variables a scientist should control when preparing liquid medium cultures. Oh. Probably the temperature? Oh. I was literally going to say temperature and pH. No, those are already them. I'm going to guess the volume of culture. Like, the volume of culture. Volume of culture. Volume of bacteria culture. Time left. Okay, yeah. Okay. Put a tick in the box next to a process that occurs in anaerobic respiration but does not occur in aerobic. Oh. Reduction of NAD occurs in both. Phosphorylation occurs in both. Substrate level phosphorylation occurs in both. Does it? Because the other one's reduction of pyruvate. Which happens. Pyruvate gets... Is it phosphorylation? Reduction of pyruvate. Okay, so that was my original answer, but I thought... I guess because the link reaction doesn't happen. Okay. Let's see. So just how speciation happened. I'm gonna guess that it is likely to do with... Is it gonna be sympatric speciation? Or allopatric speciation? Because I wrote a very good essay on one of them, but I don't remember which. Sympatric I did, which is where random mutation leads to reproductive isolation. So they can no longer produce fertile offspring. that is obviously going to lead to a prevention of gene flow from occurring, which will then lead to, over time, 2 different species. I'm probably going to wrap things up there. I think we've been live for a good, 40 minutes here. I'm happy with that. We basically have just gone over some generic stuff, but that was actually very, very helpful. I cannot lie. That has been helpful, because I did forget a lot of that. I always forget things like the Krebs cycle. So it's good to always go back over it. And I'm very happy in this instance that we did, because that was a 6-mark exam question that I likely would have got half marks on. So, very helpful. Goodbye.